even though some people think it's 9:10, they're going to walk in the door any minute now, like here comes one now. But thank you all for coming this morning. It's my pleasure to be able to introduce Frida this morning. Life is strange and it runs through some weird curves every now and again. And I've always thought the measure of a person's not how they deal with normal everyday life, whether they excel at what they do. It's what happens when things get weird and how you handle that. And you know, graduate school is kind of a strange event anyway, but things do get weird. Harry lost his way to professor a month before he defended. Talk about getting weird. Mine didn't get weird until my graduate grading was kind of weird. But my postdoc didn't get was where things got weird for me because I ran into a terrible postdoc situation and I quit without another job, thinking, I'll never work in science again. Which clearly wasn't true. So at the moment things seem intense and it seems like we're never going to survive them, but we do. And Frida is a survivor in many respects. This is her second professional career. But she is from both the Hunt and the Open Lab, as you'll see. And she's going to tell us the sort of work she did in the Open Lab for her degree. Frida.
Thank you. Thank you for coming today. And I will be presenting all the work that I've done will be from the Open Lab. And Dr. Hans for the past 6 months have guided me in writing a dissertation. And today I'll be presenting the topic of to be or not to be, the molecular mechanisms that regulate spermatogonia stem cell fate, as you can see on the Shakespeare pen. So sexual reproduction is very important for mammalian species, and it is important because it helps the survival of a species. It has a lot of evolutionary benefits, and additionally, how it helps the survival of a species and contribute to evolutionary fitness is by transmitting genetic and epigenetic information to the next generation. So for human reproductive age couples worldwide, approximately 10% of them experience infertility. And of those infertility cases, approximately half of them are contributed by male factors. And the gametes for the male is called spermatozoa. And it can be disrupted by genetic disorders, metabolic dysregulation, and also cancer therapy. And the process of producing spermatozoa is called spermatogenesis. And within male, you can find it within the testis. And here is a cartoon representation of spermatogenesis. When you take a cross-section of a male gonadal testis, you can find various seminiferous tubules. And when you take a cross-section of a seminiferous tubule, it looks like this, like a pepperoni pizza. The pepperonis are the germ cells, and the nerve cells are the sheets. And if you take a slice of this cross-section, you can see the spermatogonia population along the basement membrane, and then it goes through meiosis, becomes primary and secondary spermatocytes, then it becomes spermatids, and becomes sperm cells in the lumen. So this process within a reproductive male with a normal lifespan produce approximately 1 trillion spermatozoa throughout their lifetime. And here, the spermatogonia population is where I will focus my talk today. And within the spermatogonia population resides spermatogonia stem cells, and outside the entire germ cell population, approximately 0.03% of them are spermatogonial stem cells, a very rare population. How do we define spermatogonial stem cells? Spermatogonial stem cell is defined by the ability to self-renew and proliferate so that they can sustain spermogenesis. Additionally, they are also defined by the ability to restore spermogenesis after transplantation into a germ cell depleted recipient. Here, let me further define how they can self-renew and proliferate. The green cell is a spermatogonial stem cell. It can self-renew, produce another spermatogonial stem cell, or it can transition into a progenitor cell. This is an asingle progenitor cell. The progenitor cell then can proceed on to incomplete mitosis, here you can see a intracellular bridge connecting the A-aligned cell. These cells are transiently amplified into A-aligned cells that can be 4, 8, 16, 32, and 64 cells. And upon being exposed to retinoic acids, these cells then proceed to differentiate and continue with spermatogenesis. Within this population of spermogonial stem cell and progenitor, just these two populations, approximately 10 to 20% of these cells are spermogonial stem cells. Next, how do we discern whether spermogonial stem cells can have regenerative capacity? In the Brinster lab, they have established a culture technique where they take a donor mouse. In the Oakley lab, we use the Rosa mouse, where you take the testis of a donor, making single cell suspension, and then using Thy1 as a cell surface marker to select for germ cells. And then these cells are cultured on feeder cell monolayer in defined media, very importantly with growth factor GNF and FGF2. And for consistency, all the cell cultures when I was in the Oakley lab were done by Melissa Oakley. These cells that were cultured in the cell culture has both the progenitor and the spermogonial stem cell population. And then these cells, after culture and the cell being established, they are injected into a recipient's testis. The recipient's testis were treated with busulfan, which is an alkylating agent to eliminate the recipient testis' germ cells. After injection of the donor cells into the recipient testis, 2 months after transplantation, the recipient testes were extracted, collected, exposed to excal. And remember, the Rosa mouse, I forgot to let you know, they are constitutively expressing the LAC-Z gene, so that in the presence of excal, all those cells turn blue. So spermatogonial stem cell with regenerative capacity in the donor testis, once they reestablish spermatogenesis, all the colonies turn blue. And how we go about finding out if there's a colony or not, we take the tourniquet off, expose the testis to excal.
So that'll do it.
All right, have a good one.
You too.
Thanks, bye.
And then we carefully use tweezers to tweeze it out so they will look like spaghetti. So whenever you see a blue spaghetti, that's a spermogonial stem cell that has regenerated a colony. And so approximately 10 to 20% of those injected cells are spermogonial stem cells after you count them. So all these techniques, the transplantation is done by Dr. Oakley in the Oakley lab. So let's talk about markers. So let's just go over again. Spermogonial stem cells have the capacity to self-renew, proliferate, and become single progenitor cells, and then A-aligned progenitor cells. So there are markers that will select for, that express and select for the heterogeneous population of spermogonial stem cells and progenitor. Additionally, there are markers that will select for a subpopulation of, again, the heterogeneous population of spermatogonial stem cells and progenitor. I would like to bring your attention to PLZF, Lin28, and GFR-alpha-1. These markers will be used subsequently throughout my talk as a baseline to compare the other genes that we are looking at. So before the Oakley lab published the paper in 2011 with ID4 as a putative marker for spermogonial stem cell, there was no marker that could select for a pure population of spermogonial stem cell. Additionally, there was no marker that can distinguish the single population and the spermogonial stem cell population. So throughout this talk, I'll present to you evidence for a tool to select for the spermogonial stem cell population and also identify the progenitor and spermogonial heterogeneous population. So first, the Oakley lab proceeded in defining a molecular marker for the spermogonial stem cell population. And as I previously indicated growth factor is very important for culturing spermogonial stem cells or germ cell cultures. GDNF, glial-derived neurotrophic factor, is added as a growth factor. Dr. Oatley decided to add GDNF and also withdraw GDNF, collected the RNA, and then did gene expression analysis, and he identified ID4, inhibitor of DNA binding 4, as a putative marker or a candidate gene for regulating spermogonial stem cell maintenance. This marker, this gene, is a dominant negative transcription regulator. Additionally, it is found to be very important for myogenesis, neurogenesis, and hematopoiesis. Dr. Oatley then proceeded to compare the expression of ID4 with PLZF, which is the heterogeneous marker for progenitor and spermogonial stem cell. And in the 2011 publication, he was able to show that the red here is the ID4, green here is PLZF, and as you combine the two, you can probably see it better now with brown here. So red right here, and then green. And the combined, you can see that ID4 is a subpopulation of the PLZF expressing cells, indicating that ID4 is a putative marker for spermogonial stem cells. Next, in order to further understand cells that express ID4 and the function of ID4, and also how it is expressed within the testis, the Oatley lab had a collaborator in Penn State generating a transgenic mouse where they express the ID4 is tagged by a GFP. The back clone is then pronuclear injected into a donor egg to derive an ID4 GFP transgenic mouse. This mouse then is crossed with a Rosa mouse so that the Rosa ID4 mouse has not only ID4 GFP marking the putative spermogonial stem cells, also all the cells, again, will express the LAC-Z gene so that when transplantation analysis is performed, we can see the colonies derived by donor spermogonial stem cells. So I took the ID4 GFP transgenic mouse and collected testes from various developmental stages from day 0, day 3 after birth, postnatal day 6, postnatal day 12, to postnatal day 20 and 35, and used immunohistochemistry to stain for ID4 GFP. As you can see here, the staining for the GFP decrease and becomes more rare and less frequent as the testes mature, which is congruent with previous definition of spermogonial stem cells within the gonad. Next, we wanted to see whether ID4 GFP, the expression pattern compared to PLZF. Live imaging of day 6, postnatal day 6, show that the ID4 GFP is definitely expressed in single spermatogonia, and by the time in a mature testis on day 35, postnatal day 35, it becomes a rare population. Additionally, I was able to show that red is PLZF. Again, remember it is showing both progenitor as spermogonial stem cell population, and progenitor cells are connected by an intracellular bridge because of incomplete mitosis. And as you can see here, the GFP population is in the single cell population, whereas PLZF marks progenitor, which is connected by intracellular bridge, indicating that the ID4 GFP is a tool to identify spermogonial stem cells within the testis. With that in mind, Melissa generated an ID4 GFP Rosa mouse culture using Thiogon again to select whole germ cells, culturing it on beta cell monolayer with GDNF and FGF2. Using fluorescent flow cytometry, we were able to discern that approximately 10 to 20% of those cells are GFP positive, similar to previously shown results where spermogonial stem cells are approximately 10 to 20% of the culture population. Next, Melissa took the cells and separated them with fluorescence activated cell sorting, the GFP positive putative spermatogonia stem cell population, and the GFP negative, the progenitor population, transplanted them into germ cell depleted recipient. 2 months after transplantation, these cells, these testes are taken and exposed to X-GAP. And as you can see here, ID4-GFP had an overabundance of regenerative capacity, whereas the GFP negative population has little to none of regenerative capacity, indicating that ID4-GFP is a tool to select for an enriched or pure spermatogonia stem cell population. This is very powerful because then we can use this tool again to identify the genes that regulate spermatogonia stem cell maintenance. To do these, the spermatogonia stem cells are separated using the ID4-GFP. So the ID4-GFP positive population are separated from the negative progenitor population, RNA sequencing, and the RNA sequencing analysis show that with top hats and also the tuxedo package show that there are 11 genes that are upregulated or only expressed in the spermatogonia stem cell population. These genes include genes that are important for transcription, high RNA metabolism, cell growth and metabolism, and also intracellular signaling. Looking at these 11 genes, 3 of them become extremely interesting to me, which include UTF1 and enolase 1 and 2. So from now on, after this section, I will talk about my investigating of these 3 genes. So to summarize, so far, is that the OP lab has generated an ID4-GFP transgenic mouse as a novel tool to select for spermatogonia stem cells. ID4-GFP expression is found to localize in single spermatogonia, a rare spermatogonia stem cell population. And only ID4-GFP positive cells has regenerative capacity. Additionally, the 11 genes that were upregulated or only expressed in spermatogonia stem cell population is putative genes for regulating spermatogonia stem cell maintenance. This includes UTF1 and also enolase 1 and 2. So now we'll shift gear and talk about UTF1. So what is the biological relevance of UTF1? UTF1, when I looked into the literature, is expressed by both embryonic stem cells and iPS cells. So UTF1 is also known as undifferentiated transcription factor 1. Although its name is called transcription factor, everything that it does has nothing to do with transcription factor. It is actually a chromatin-associated protein. It regulates voice genes, which are both H3K4 trimethylated for activation and H3K27 trimethylated for repression. Additionally, when researchers previously published data suggest that UTF1 deficiency or knockdown has delay or block in ESL differentiation. So let's talk about what voice genes are, because I'm sure a lot of you are wondering what it is. So voice genes are both H3K4 trimethylated for activation, which will inhibit DNA methylation, which then the genes are activated. H3K27 trimethylation is for repression because it inhibits RNA polymerase from binding, so therefore the genes are repressed. So with both of them being trimethylated, these are called voice genes. And these voice genes has been suggested to be very important during development. And either of these marks are resolved so that the genes can either be activated immediately or be repressed upon differentiation. UTF1 has been suggested to bind to the voice genes to regulate either overexpression. If it's overly activated, it will then tag the mRNA for degradation, or it also prevents over-repression, so over H3K27 trimethylation, so that it regulates further the voice genes. So So additionally, back in 2008, the Roy's lab has shown that UTF1 is expressed in the single spermatogoneous population, but they have not performed functional studies since. So for me, even though it's expressed in rat testes, I thought it would be very interesting to examine it in the mouse testes and also explore the functional side of UTF1. To do this, the spermatogonial stem cell culture was separated into the GFP positive spermatogonial stem cell population and the GFP negative progenitor population. These populations are then checked for gene expression analysis with both real-time quantitative PCR and Western blot. So I haven't shown the real-time quantitative PCR here, but the data confirms the RNA-seq data where UTF1 is upregulated in the spermatogonial stem cell population. And you can see here is a representative image of a Western blot showing that spermatogonial stem cells, UTF1 is definitely, protein is enriched in that population compared to the progenitor population. With 3 biological replicates, I was able to show that there is definitely a difference in protein abundance in the spermatogonial stem cell population, which confirms that UTF1 is a putative gene for spermatogonial stem cell maintenance. Next, I wanted to examine whether UTF1 expression is localized to spermatogonial stem cell. To do this, I used the ID4GFP mark as a tool to compare UTF1 expression within testes. I collected testes from postnatal day 3, 6, 12, 20, and adult. Immunostained them for both UTF1 and ID4GFP. And as you can see, in a immature 3-day-old testis, both UTF1 and ID4 are colocalized to the same cell. And as the testes mature, spermatogonial stem cells become a subpopulation of the UTF1 positive population. In an adult, you can see that it becomes very rare to find both seen for these cells. And because there is a UTF1 positive ID4GFP negative population, I wanted to examine the expression pattern of UTF1 compared to other progenitor cells. So I used LIN28 as a marker to compare the UTF1 expression with LIN28 expression. I collected testes from the ID4GFP transgenic mouse at day 3, 6, 12, 20, and adults. Stained them for both UTF1 and LIN28. As you can see, in the mature day 3 testis, they are colocalized in the same cells. As they mature, the red as UTF1 became a subpopulation of the heterogeneous progenitor population as marked by LIN28, indicating that UTF1 is expressed by both spermatogonial stem cell and a subpopulation of progenitor, which is here. So this is interesting because UTF1 is a regulator of voice genes, and it suggests that UTF1 can regulate voice genes within spermatogonial stem cells. Next, I shift gear to examine whether UTF1 because it's expressed in spermatogonial stem cell, does it actually regulate spermatogonial stem cell maintenance? To do this, I treated ID4GFP culture with both UTF1 siRNA and control siRNA. This is to examine whether depletion of UTF1 would inhibit spermatogonial stem cell function. After treatment, these cells are transplanted by Dr. Oli into recipient's testes. 2 months after transplantation, these testes are collected, exposed to XGAP. And as you can see here, control versus UTF1. I then twisted open, counted the colonies of regenerated spermatogenesis. This is the control, and UTF1 siRNA, which is translucent deficient in UTF1, shows that there is a decreased amount of regenerative capacity, indicating that UTF1 is important for spermatogonial stem cell maintenance. Knowing that UTF1 is important for spermatogonial stem cell maintenance, I wanted to know how UTF1 is expressed and how its expression is regulated in spermatogonial stem cell. looking into the literature, I find that in embryonic stem cells and iPS cells, UTF1 is activated by SOX2. SOX2 is a sex-determining region-wide FOX2 protein. It binds to the promoter region of UTF1 and to activate it. Additionally, in mouse testis, a germ cell-specific ablation of UTF1 disrupts spermatogenesis. And as you can see here, instead of a pepperoni pizza, we have a cheese pizza with no germ cells. So for me, it became important to understand whether SOX2 binds to the promoter region of UTF1 to activate, to maintain spermatogonal stem cells. So first, I wanted to know if SOX2 is also upregulated in the ID4-GFP positive population. Again, I used the GFP positive spermatogonal stem cells and the GFP negative progenitor cells. Here's a representative image of the Western blot showing that the SOX2 protein is definitely enriched in the spermatogonal stem cell population in compared to progenitor population. And with 3 biological replicates, it shows that SOX2 protein is upregulated in the spermatogonal stem cell population. So next, knowing that SOX2 colocalizes, they exist in spermatogonal stem cells, well, does it bind to the promoter region of UTF1? To do this, here is a representative image of the UTF1 promoter region. And this is in purple is the binding sites for SOX2. And I designed both forward and reverse pattern for this region for a chip analysis. Additionally, I also designed negative control primer for recognizing another region that's not here to make sure that my pull downs are for this region. Just brief, for those people who don't know what a chip analysis is, just a brief summary of what it is. I take the spermatogonal stem cell culture, cross-link them, so that the proteins that attach to the DNA sonicate it to collect the DNA protein complex, and then immunoprecipitate it either with IgG as my negative control, input DNA, everything, and then also specific SOX2 chip, which is to pull down the SOX2 DNA complex. And then I purify the DNA, and then using the primers that I designed, both the UTF1 promoter region and also the negative control region. And here, the results from spermatogonal stem cells show that SOX2 definitely binds to the promoter region of UTF1, and I use embryonic stem cells as a positive control. Additionally, in the negative control, you can see that there is no primer, indicating that SOX2 does bind to the UTF1 promoter region. So just because it binds does not mean that it activates. Next, I wanted to find out if SOX2 actually activates UTF1, and therefore helping maintain spermatogonal stem cells. To do this, I treated the spermatogonal germ cell culture with SOX2 siRNA and control siRNA. This is to deplete the SOX2 abundance and see whether the transcription and also protein abundance of UTF1 is affected. Here is representative quantification of 3 different biological replicates of SOX2 treated siRNA in the top. Right here, and it shows that SOX2 siRNA has depleted transcript abundance. At the same time, SOX2 transcript abundance decreased, UTF1 transcript abundance also decreased. Next, I examined the protein abundance. With the SOX2 siRNA treated, there's decreased amount of SOX2 protein and also decreased amount of UTF1 protein. And this is a quantification showing that SOX2 siRNA treated population has decreased SOX2 protein abundance and also decreased UTF1 protein abundance. So in summary, for this UTF1 portion, I was able to show that SOX2 activates UTF1 to maintain, avoid spermatogonal stem cell maintenance. And spermogonial stem cell in a subpopulation of progenitor expressed UTF1. And it is very important for UTF1 expression so that spermogonial stem cells can restore spermatogenesis. And again, SOX2 activates UTF1 transcription within spermogonial stem cells. And as you can see here, UTF1 is a putative marker for the a single spermogonial stem cell population and a single progenitor population. So the next step will be to identify the poise genes to determine whether there are poise genes in spermogonial stem cells and progenitor. And also the UTF1, how it regulates those poise genes in spermogonial stem cells, and also the chromatin modification during the transition of spermogonial stem cells to progenitor, which genes are resolved. And those will be very important to understand spermogonial stem cells development and spermatogenesis in general. Next, with the last portion, shifting gear to enolase 1 and 2. So why I chose enolase 1 and 2? Because it is involved in glycolysis, and glycolysis is a preferred metabolic pathway for most stem cell type. Additionally, when I looked into the 25 genes that are upregulated or only expressed in the progenitor population, there are genes that are associated with mitochondria, indicating the transiently amplifying progenitor population has increased amounts of progenitor, which would support my hypothesis that enolase 1 and 2 is important for spermogonial stem cells. So again, to go over some biological relevance of enolase 1 and 2, for a lot of you, picture is much better than words. As you can see here, enolase 1 and 2 is the penultimate step in glycolysis. Its product is PEP. And then also, mitochondria is important for oxidative phosphorylation, and these genes are associated with mitochondria in general. So a lot of you saying, well, enolase 1 and 2 is not the last step of glycolysis, and also spermogonial stem cell can and might, no one knows yet, but the Sertoli cell provide a lot of pyruvate. So why enolase 1 and 2? So I looked again into literature. Aside from being glycolysis enzyme, enolase 1 is a jack of all trades. It is important for hypoxic stress response. It binds to plasmin and plasminogen. The mRNA for enolase 1 can be started at another codon to translate for the MVP1 protein, which regulates c-MYC transcription, and that gene is very important for proliferation of stem cells. And then it's also known to be associated with neurotrophic factor. It is also associated with the centrosome. It's important for heat shock protein. And then it also binds to F-actin and tubulin. So it is definitely a jack of all trades. So first, before examining anything, again, I wanted to confirm the RNA-seq data. To do that, proteins and RNA were collected from both the spermogonial stem cell GFP positive population and the progenitor cell GFP negative population. This is a representative image for a Western blot examining the protein abundance of enolase 1 and 2. And as you can see here, is the enolase 1 and 2 in spermogonial stem cells versus progenitor. And here is 3 different biological replicates of the protein analysis in Western blot. As you can see, enolase 1 and 2 are enriched in the spermogonial stem cell population. Next, just because it's upregulated does not mean it's performing anything important, so I wanted to examine whether enolase 1 and 2 is important for spermogonial stem cell maintenance. To do that, I treated the spermogonial stem cell culture with both enolase 1 and 2 siRNA and also control. This way, both 1 and 2 will be transiently reduced so that there will not be compensating effect. These treated cells are then transplanted into germ cell depleted recipients tested by Dr. Obey. After 2 months, these testes were collected, exposed them to X-scale, here's a control, and this is the enolase 1 and 2 treated with siRNA. Analysis, choosing it apart, counting the recolonization. This is 3 different biological replicates showing that control is here. Enolase 1 and 2 treated cells definitely has a depleted amount of restored spermatogenesis, indicating that enolase 1 and 2 are important for spermatogonal stem cell maintenance. So, to summarize, enolase 1 and 2 is important for regulating spermatogonal stem cells, and it is upregulated in the spermatogonal stem cell population. And again, it is important for restoration of spermatogenesis by spermatogonal stem cells. So to summarize everything that I've talked about today, just kidding. The future directions for the enolase 1 and 2 portion. So we need to determine if enolase 1 and 2 are upregulated by spermatogonal stem cells, does it actually augment glycolysis within those cells? And whether spermatogonal stem cells' preferred metabolic pathway is glycolysis or something else. This will help us to study the bioenergetics for spermatogonal stem cells and progenitor cells. And next, also determine whether enolase 1 perform a non-glycolytic function within spermatogonal stem cells. Does it regulate c-MYC transcription? Does it bind to plasminogen? And does it help hypoxic stress response? So now is definitely the overall summary of everything. The OB lab was able to generate an ID4GMP transgenic mouse, and it was used as a tool to select for a total population of spermatogonal stem cell. UTF1 was identified as a marker to mark the spermatogonal stem cell single population and also the asingle spermatogonal population, the progenitor population. These tool and marker can be very important for analyzing and understanding the transcriptome and also the epigenome for these cells. The epigenome is very important because they regulate the cell's transcriptome, the gene expression, and also will understand each steps, the methylation, and also other epigenetic marks that are important that regulates the transition between spermatogonal stem cell to progenitor. Next, enolase 1 and 2 can help us understand the bioenergetics that are important for spermatogonal stem cell maintenance and also the transition to progenitor cells. In total, these findings are going to contribute to helping diagnose and also treating male infertility. And of course, it will contribute to the reproductive biology field. So I would like to take this time to thank Dr. Hunt for helping me in the past 6 months with my journey. And additionally, I would like to thank Dr. Oli for providing the opportunity to do some great science. Melissa for providing an abundance of cells and also other support. And then my undergrad that has helped with so much because she gave me 4 hands, 2 pair of hands. And also all other members within the OB lab that are current and past that has helped with the journey. Additionally, I would like to thank my current and past committee member who has guided me throughout the entire process of getting my PhD. Very importantly, the graduate committee and all the faculties and all my friends and Kelly McGovern for helping me through all the hurdles and getting my PhD. The Maisa I sacrificed, and also the funding from that made everything possible. Without funding, nothing is possible. So I would like to thank Dr. Oli for getting the funding and also the training grants. And additionally, I would like to thank my musical family because science is tough, and they were there, and I will go into rehearsals, reading scientific article during the rest, and they'll poke at me when it's time to play. So it's really nice. My boys in the WSU rowing team, who were there through thick and thin. Justin, putting up with me, he told me last night, and he actually did, was able to start reciting this presentation. So he could very well substitute for me and stand up here. My dad, my family, but especially my dad, people had, what is that called? When they go to bed, they have bedtime stories. I had science stories. So since I was a little girl, we talked about science. And he looks like a hipster here. That's why I love his picture. My best friend in science, Phoebe, when she left, it was really hard. And my girls who helped me, and Nicole. And then also there's Tim, Sean, and Karen right there who support me through everything. I don't have a picture, sorry. And then Simon. He is my kindred spirits for science, music, and food. And he passed away, so I can't thank him, but I would like to thank him. So, that's it.
Question?
Yes.
I have a generic question about stem cells. Is the transition of a stem cell to a more differentiated state, is it typical that a stem cell uses glycolysis more frequently, and then when it moves on, it goes to the mitochondria as an energetic source? I'm just wondering if that's
So
a typical
for
thing.
embryonic stem cells, definitely, and iPS cells, they prefer the glycolytic pathway. It has also been shown, there was actually from a PI in Boston, they were able to show that glycolysis is a preferred pathway for some neural stem cells and also some muscle stem cells and hematopoietic stem cells. But, you know, it's different stem cells are different. That's why it's still important to understand whether glycolysis is the preferred pathway. And it's kind of weird that enolase is the penultimate step, not the last step. So the enolase might be upregulated for glycolysis, but it also might have other functions. Catherine?
So I'm really curious about your intracellular localization of UTF1. It looks cytoplasmic in a lot of cells.
It does.
And then nuclear in a couple. So do you think that perhaps it's subcellular trafficking, maybe part of its mechanism?
That might be. It's very interesting. That's a very good point. So that will be important to find out for the function. You know, further analysis for the function to understand whether it has actually a trafficking element to it, and whether it functions outside and also inside, whether when it's binding to the chromatin, it goes into the nucleus, and then when it's not binding, it's outside in the cytoplasm waiting to do something.
So I was wondering whether if you noticed that with markers with the different other undifferentiated markers, whether it was more nuclear in IDF ones, or whether it was more cytoplasmic when you localized
So
LIN28.
that is interesting too, because when I use different fixation and also antigen retrieval method, they localize to different parts. So it might be artifacts of fixation, but I know that LIN28 sometimes do that, from what I have seen.
Yes.
So
And PLZF does it too.
yeah, so I don't know if it's just an artifact of, because using UTF1, it was really finicky, and there was only one way of treating, fixing testes, and one way of doing antigen retrieval that would make both, LIN28 and UTF1 work. And those were the two that actually worked together, and then that's it. Yeah, so that's an interesting question. So, yes?
I have a question that's sort of an extension of Dr. Heinz's question. So it's about your RNA sequencing. Did you see any other genes in your 94GFP positive sample that would point you towards energetics? Like were there more glycolysis genes upregulated besides enolases, or other potential pathways that were found regulated?
So Travis's question is whether there are other genes that I saw in the RNA-seq data that would point toward glycolysis. The answer is no, not within the stream that we use. But it doesn't mean that those genes are not expressed. So the genes are there and the proteins are there because all cells is ubiquitously expressing the glycolysis pathway. But that's why it's important to further examine it and see if the products, the byproducts like PEP from enolase is upregulated. If it is, then we know it's probably performing glycolysis. If it's not, then it's probably one of the other functions. Yes?
So I was going to build on these guys' questions again. So I think Travis asked you about things that are down-regulated in this population. So how many genes, you said there were 11 up-regulated, how many genes are down-regulated in the population compared to the progenitor cells?
So we only looked at the genes that are upregulated or only expressed. So the in-between parts are the common genes. So we didn't actually see down-regulated genes. So we're only selecting for upregulated.
And then to extend John's question, I mean, stem cells in the skin are fairly well identified and characterized. So is there overlap between the genes that you're looking at here and specifically skin cells? I'm biased about skin.
That's a very good question, and it would be nice to, take the information and compare them. So that would definitely be a future direction that would be very interesting to look at the common genes that are expressed by other tissue-specific stem cells and whether they have similar. But I know that from looking, UTF1 is only in embryonic stem cells, iPS cells, and spermatogonia stem cells. And I couldn't find it, at least in the literature, in other cell types. That's why it's very interesting that it's binding to voice genes. Yes?
So UTF1 is upregulated early in the ID4 positive cell. Is it expressed in? I guess what I'm wondering is, it goes away not long after ID4 signal starts to go away.
Mm-hmm.
And if it's regulating chromatin structure, but it's being depleted soon in differentiation, how long do you think what's the effect of changes in chromatin structure on gene expression? Can changes in chromatin structure rapidly account for changes in gene expression? I'm just asking about the time frame for depletion of UTF1 and transcription.
So let me see if this is what you asked. So you're asking that ID4-GFP is those cells express UTF1, and then once it starts differentiating and becoming progenitor cells, UTF1 is then downregulated and depleted. And you're wondering the chromatin changes that occurs that regulates that developmental pathway. So that's a very interesting question, and a lot of people have actually been trying to publish data. So Hamid from, I think, Colorado or Utah, she actually took the entire Thy1 positive population, which is progenitor and spermatogonia stem cells, comparing to other stages of spermatogenesis to map out all the voice genes. But she didn't actually go further. It's a great step forward. It's absolutely phenomenal because we can see the developmental pathway as all the chromatin are being resolved and go forward in developmental. And then they also see that there's a subset of voice genes that are expressed in embryonic stem cells that carry through in all those cells. But what they didn't do is, they didn't have the tool, is to separate out that Thy1 population into both progenitor and then spermatogonia stem cells, then compare the chromatin changes in that those subpopulations. And so it'll be interesting to take out the GFP positive, UTF1 positive population, find out what's the chromatin is doing there. And then for me, that in between section where GFP positive is negative, but UTF1 is still positive, that's very interesting to understand what that population is doing and whether those cells can go backwards, manipulated genetically to go backwards, become spermatogonia stem cells, and then the negative for both GFP and also negative for UTF1. And those are they absolutely going forward and no way going back. So then we can understand the epigenome changes that regulates all the developmental stages of spermatogenesis. Does that answer your question? Kind of.
All right. If there are no more questions, we'd like to continue to help you. Faculty are welcome to join us right after this. Trainees, don't forget, Sean's talking 210. That will be awesome. Don't forget. But join me in thanking